electron transport chain happens in a crystal of the mitochondria. So in aerobic prokaryotes, it happens in the plasma membrane. So we have a series of reaction that happens here. So, and we have the nicotinamide adenine dinucleotide will convert to, so we have, so this process we see here is actually the process which is the end of the reaction process of respiration. So aerobic prokaryotes it's got plasma, is up in the plasma membrane. So we have a series of carrier molecules here. So what do we mean by a series of carrier molecules? The meaning is every section we have a carrier protein which is going to carry to another level. So a pass energy-rich electrons, the energy-rich electrons, which are energized electrons, will successfully move one another to the transport chain. So there is a complex area of proteins and a cytochrome. So this would help the movement of these electrons. Proteins with heme group with central ion molecule when central ion atoms would also be involved in the reaction. So what happens in the electron transport chain? It receives electrons from NADH and FADH2. So it produces ATP by oxidative phosphorylation. So oxidative phosphorylation is a process of production of ATP. So oxygen final electron acceptor combines with hydrogen ion to form water. So mind you, oxygen would be sent into the system, and you've got hydrogen ion in the system. So that leads to the production of water molecule. So now let's talk about the cycling carriers. So what is the fate of hydrogens in the process? So hydrogen from the NADH deliver enough energy to make 3 ATPs. So energy, because we told you before, NADH is just an energy carrier. It's a coenzyme that helps to transport energy. So this energy carrier coenzyme would have to deliver enough energy to make 3 ATPs. So those energy from FADH2 have only enough for 2 ATPs. So that was the reason I told you before that NAD plus is usually wanted to be used and reused again. So FADH2 would only have enough to have, it's got only very little energy that could only produce 2 ATPs, while NADH could produce 3 ATPs. So spent hydrogen will combine with oxygen. So the spent hydrogens will combine with oxygen. So we recycle. What do we mean when we say recycling of coenzyme increases in efficiency? So the coenzymes, when we recycle them, they have to increase the efficiency of the process. So once the NADH delivers the hydrogen, it returns back to NAD plus. So to pick more hydrogen. So I told you before, we have abundance of hydrogen in the intermembrane during the process intermembrane of the mitochondria. So it goes to pick another one. So, however, hydrogens must be combined with oxygen to make water. So oxygen is not present. So what happens is NADH would not release this. So it's only going to release hydrogen ion when oxygen is present. So it's no longer recycled back to NAD plus. So the point is, it is a presence of oxygen that will make the release of hydrogen ion and going back to pick another one. But once there is no oxygen in the system, it's not going to be able to go back and recycle the same thing again. So the electron transport chain complexes would not pump hydrogen ion from the matrix. Matrix is the open space of the mitochondria into the intermembrane space of the mitochondria. So it pumps the hydrogen ions. So these hydrogen ions has been accumulated from the beginning of the process. So it's been accumulated since every breaking down would produce this. Every time there is an oxidation process, is going to lead to the production of hydrogen ion. So hydrogen ion therefore becomes more concentrated in the intermembrane space. So this will create what we call an electrochemical gradient. So once you have more of hydrogen ion, which has been pumped through the water, through the hydrogen ion pump into the wall, into the intermembrane space. So it's going to lead to a high concentration of hydrogen ion in the what? In the intermembrane space. That is to say, the space between the inner membrane and outer membrane of the mitochondria. So the ATP synthase would allow hydrogen ion to flow down its gradient. So it's going to allow hydrogen ion to flow down its gradient. So because it needs to produce more ATP. So the flow of hydrogen ion drives the synthesis of ATP from ADP. So and in inorganic phosphate by ATP synthase. So it's going to what? It's going to drive it. And this process is called osmosis. ATP production is linked to the establishment of hydrogen ion gradient. So since you have a gradient, high gradient of hydrogen ion in the intermembrane space, so ATP will move out of the mitochondria. So ATP moves out of the mitochondria and is being used for cellular actions in the cell. So it can be broken down to ADP and inorganic phosphate, and these molecules are returned to the mitochondria for more production of ATP when they need to produce it. So this is a description of it. So in this process, we can see how we can see how there is a net movement of hydrogen ion out of the wall from this from this space into the wall intermembrane space. So it's being moved from the matrix into the intermembrane space, and very little is actually coming in, but not that much. So this would lead to the what production of. So this is a matrix here, and this is the intermembrane space here. So the pumping of this hydrogen ion will lead to the production of more ATP, which we talked about before. So now what happens is the energy yield from glucose metabolism, the net energy, the net yield of glucose from glycolysis is 2 ATP. So the net production of ATP from citric cycle is 2 ATP. But from the electron transport chain, we'll have 32 or 34 ATPs. So what is energy content of 2 ATPs? So the reactant, which is glucose, is got 6 kilocalories. So the energy yield is 36 ATP, which is 2630 kilocalories. So the efficiency is only 39%. So, and the rest of the energy from glucose is lost as heat. So this is exactly what we need to know. Glucose has got 686 in the beginning. So, and it produced a total of 36 ATP. So that is equals to 363 kilocalories. How did we get this? We can calculate it by what we did before. 14.1 was equals to 2 ATPs. So when you multiply it, you're going to get this. So only 39% is efficient and the rest is lost as heat. So now this is a cytoplasm. It's trying to bring all the reaction again from the beginning. It wants to account for the energy yield per glucose molecule breakdown. So here, 2 ATP is being used to break down the glucose to pyruvate, and also leads to the production of 2 NADH. So then that enters the electron transport chain. You have 4 to 6 ATPs being produced. So this is in the production of 2-acetyl-CoA. So why does this say 2 or 4? Because this procedure will happen 2 times. So, and it's going to produce 2 ATPs. So acetyl-CoA, 2 NADH is been used into the system, and 6 ATPs is been produced. So, and then also the citric cycle, it produces 6 NADH. So 6 NADH will lead to the production of 18 ATPs, and 2 FADH2 will lead to the production of 4 ATP. I told you before that 1 NADH would produce 3 ATPs because it's more energized than FADH2. And 1 FADH2 will produce only 2 ATPs because it doesn't have that much energy to produce more than that. And mind you, the procedure here, before you get to the citric cycle, produce only 2 NADH. And so this is why, so if it's going to produce 2 NADH, so just multiply, because every 1 NADH will produce 3 ATPs. So that is why you've got 6 here. And 2 NADH here will produce 4 or 6. Why did it say 4 or 6? Because mind you, you used 2 before. So if the glycolysis procedure actually uses 2 ATP, so it's not going to be 6, it's going to be 4. So because you're going to subtract 2, which has been used to break down glucose from this. So, but the rest, that's the reason why the rest have a consistent production. So if you sum all of this together, it's going to give you what? A total of 32 or 34 in the what at the end of the reaction. So the reason it's saying 32 or 34 is because some textbook will not talk about the 2, which has been used in the breaking down of glucose. So water molecule being sent into the system with 6 oxygen. So 6 oxygen into the system will combine with a hydrogen ion to produce for you 6H2O. So that is the reason why it says what 36 or 38 on a total, because you've got 4 total here. So the net you have here during the process of glycolysis is 2. And during the process of citric acid, you've got another 2 as well in the mitochondria. So this is only done in an electron transport chain. So this is the one produced. So this is the reason why 4 plus 32 or 36 or 34, 2 or 34 will give you a total of 36 or 38 ATPs, theoretically. So now let's move on to the next stage. Now let's move on to the next stage, which is metabolism. Metabolism, food is a process of breaking down food. So it's a source of energy-rich molecules, carbohydrate, fat, and proteins, the source of food. So these, when you want to degrade, when you want to break down this food nutrient, it's called a degradative reaction, which is called catabolism. So it breaks down the molecule to smaller components, which the body can use. So, and in most cases, it tends to be exergonic. It tends to release energy. So while the synthesis reaction, which is the production of something, is called anabolism. So that builds molecule these tend to be endergonic. That is to say, it would use energy before the production of something would happen. So now take a look at this metabolic pool, which you see here. So protein. So protein here, they reversibly convert to amino acid. So protein is irreversibly, that is to say, protein can become amino acid, and amino acid can also become protein. So this will lead to a citric acid cycle. So that would lead to a citric acid cycle, so which is going to release ATP. And finally, it's going to lead to electron transport chain reaction, which would also release ATP. So the carbohydrate reaction, because in this food, we've got protein, we've got carbohydrate, and we've got fat and oil. So all of these things will be broken down, will go through different reaction in the metabolic procedures in the body. So the carbohydrate, they reversibly convert to glucose, and they do carry out the process of glycolysis. So they release ATP. They lead to the production of acetyl coenzyme A. So then, which should later on lead to what? Citric acid production, citric acid cycle, which will produce ATP. So, and it's going to finally lead to what? Electron transport chain reaction. So what about fat? So the fat would branch to either fatty acid or glycerol. So carrying the glycerol procedure would go through the process of glycolysis to release ATP, and the fatty acid would go through the process, will lead to the production of acetyl-CoA. So then it's going to lead to citric acid cycle, releasing ATP, which will lead to electron transport chain, producing ATP. So what does this picture here, what does it tell? It tells you that every food in the body, every food you take into your body, usually go through a different breaking down procedure, metabolic procedure. So the protein can either become a protein or amino acid, and it's going to go through the process of glycolysis. Then it goes through the process of acetyl-CoA. Then it goes to the process of citric acid, then which is going to produce ATP, and also electron transport chain to produce ATP. So the carbohydrate will go through glucose, through glycolysis, acetyl-CoA, citric acid. It produces ATP here, produces ATP, produces ATP. You will see more ATPs have been produced when you break it down carbohydrate. Also, when you break it down fat, it does produce more ATP for you. So glucose is broken down in cellular respiration, that is catabolism. Fat breaks down into glycerol and free fatty acid. Amino acid will break down into carbon chains and amino acid. There is a procedure called deamination, which is the removal of NH2 from amino group. So this would take place in the liver. So this would result in poisonous ammonia, NH3. So this poisonous ammonia will be converted to urea, which will be removed from the body through the process of kidney filtration into the bladder. So different outgrowth from the amino acid are being processed differently. So if you have different outgrowth, the procedure of processing it is not the same. So the fragment will enter the respiratory pathway at many different points. So the meaning is the fragments you have will go through the respiratory pathway, not at the same point. So what about anabolism? All of the metabolic compounds are part of the metabolic pool. The intermediates from the respiratory pathways can be used for anabolism. So what is anabolism? Synthetic reaction of anabolism. So carbohydrates would start with the acetyl-CoA. So basically it reverses the glycolysis procedure, but in a different pathway. Fat would produce G3P, and it's going to convert that to glycerol. Acetyl group are being connected in pairs to form fatty acid. So, and anabolism, which is in protein. So what happens, there are more, we have 20 different amino acids which we produce. Some amino acid like 11 can be synthesized by adult humans, and the 9 of it cannot be synthesized. So they need to be produced. So they need to be given to you through some other process. So the essential amino acid, so they must be present in your meal. So, but this ones, the 11, they can be synthesized normally by the body, but this would have to come through the meal you take or must be present in your diet. So now let's talk about the energy organelles, which we went through in the procedure in all the processes we've talked about. So what is the similarity between photosynthesis and cellular respiration? So the similarities is they both use the membrane. So chloroplast's inner membrane forms a thylakoid, while the mitochondria's inner membrane forms a cristae. So what about the electron transport chain? ETC is located on the thylakoid membrane and a cristae. So the thylakoid membrane has the ETC and the cristae has it for the mitochondria. So in photosynthesis, electrons passed to ETC were energized by the sun. And in the mitochondria, electrons energize electrons. So in the mitochondria, energized electrons were being removed from the glucose. So the meaning is the electrons which become energized are being actually removed when the glucose is being broken down. So that is why that's the difference between the electrons energized in the two processes, and that's why you see a lot of electrons being energized in the procedure. So in both the ETC, they establish an electrochemical gradient of each hydrogen ion with the ATP production by chemiosmosis. So they have a gradient of hydrogen ion, which is a lot of hydrogen ion being produced in both processes, which actually led to the production of ATP in the procedure called chemiosmosis. So the enzyme in chloroplast stroma has what we call Calvin cycle enzymes, but the mitochondria, the matrix, contain enzyme of citric acid cycle. So, and what about the flow of energy? Energy flow from the sun through the chloroplast to carbohydrate, and then through the mitochondria to ATP molecules. So this is the flow of energy: sun, chloroplast, carbohydrate, then mitochondria to ATP molecules. So this flow of energy maintains the biological organization at all levels, from molecules to organisms to the biosphere. So energy is lost with each chemical transformation. Eventually, also the energy captured is lost. and all life dependent on solar energy input. So every life, because it's actually started, what entered the respiration cycle was actually from the photosynthesis, it was actually from the solar energy. So the chemical cycle within the natural system, chloroplasts produce oxygen and carbohydrate, and which are used by the mitochondria to generate energy for life. Chloroplasts and mitochondria, they both allow energy flow through organisms and permit chemical recycling or chemical cycling. So this is a photosynthesis versus cellular respiration in a nutshell. So in cellular respiration, glycolysis is a conversion of glucose to pyruvate and the creation of reduced form of NADPH. So NADPH is actually being produced. NADH is actually being produced in respiration. But the point is in photosynthesis is NADPH. Okay, let me talk about this and don't compare that way. So let me talk about respiration first, and I'll talk about photosynthesis later on. So it forms it forms NADH with 4 ADP converted to 4 ATP. So it creates what 2 ATP and 2 ADP in a process with a net gain of 2 ATP at the end of the reaction. So that is a glycolysis stage. So now the preparatory stage, it creates a reduced form of NADH. So when I say it creates a reduced form, that is to say it converts NAD plus to NADH. So it produces 2 ADP and 2 ATP. So the citric acid cycle is a place where you have the flavin adenine dinucleotide being produced, and also produce the nicotinamide adenine dinucleotide. So electron transport chain that creates 32 or 34 ADP and 32 or 34 ATPs. So in the crystal, what happens is oxygen converts to water with ADP converting to ATP. So, and also in the matrix, you will see glucose is being converted to carbon dioxide with NAD plus converting to NADH. So, and in solar energy is absorbed by thylakoid, as you can see here. So if it is being absorbed by the thylakoid membrane in the presence of water and gives out what NADPH and ATP. That's in the light reaction. The NADPH and ATP with a carbon cycle reaction involving carbon dioxide and glucose will give out ADP with a phosphate and NADP plus, which would help again in the light reaction. So the thylakoid membrane, in it, the water has been converted into oxygen with ADP converting into ATP. So, and also we in the stroma region, so carbon dioxide is being converted to glucose with NADH converting to NADP, NAD plus. So NAD plus. So this is exactly the comparison between the two. So you would say this happens in the stroma, this happens in the matrix. This is NADPH being reduced to NADP plus, but this NAD plus is being reduced to, this has been oxidized, sorry. So it's been reduced to NADH. So CO2 is being converted to CH2O, and this is CH2O is being converted to CO2. So an H2O has been converted to O2, and O2 has been converted to H2O. So this happens in the thylakoid membrane, this happens in the crystal, this happens in the stroma, and this happens in the matrix. So this concludes the long lesson of photosynthesis and cellular respiration. I hope you enjoyed the series of the lesson. I wish you a nice day.